The title of our webinar today is Standardizing Indoor Air Quality Measurements and Assessments. The In-Child Health Standard Operating Procedures, SOPs, and Lessons Learned. Next slide, please. Before I start, I would like to acknowledge the In-Child Health team behind this work. Our coordinator is Heidi Salonen from Aalto University in Finland, and we have partners from several European countries and associated partners from Switzerland and Australia. And of course, we acknowledge the funding through the Horizon Europe Research and Innovation Program. Next slide, please. So I will start with a brief introduction on the need for standardization. Next slide, please. Yeah, thank you. We all know that indoor air quality is important, and that we therefore have to measure and assess it accurately. We spend the large majority of our time indoors, and air quality can be worse indoors than outdoors. That can be related to emission sources in the indoor environment and to a concentration effect where there is little air exchange with the outdoor environment. As a consequence, indoor air quality can have effects on our health, well-being, and productivity. This can become especially critical for vulnerable groups such as children who we would like to protect. Next slide, please. Indoor air quality is determined by a number of parameters: physical, chemical, and biological ones. Temperature and humidity are important parameters. Then particles in different sizes, black carbon, and on the chemical side, various gases, such as carbon monoxide, carbon dioxide, volatile organic compounds, and semi-volatile organic compounds. Natural toxins are chemicals that can be produced by microorganisms, and which of course can be a problem in themselves, including bacteria, virus, and fungi. Fungi can form mold. There can also be problems with dust mites, fleas from pet animals, and other insects. Next slide, please. Indoor air quality is, of course, the central topic in the In-Child Health project. The full title of the project is Identifying Determinants for Indoor Air Quality and the Health Impacts in Environments for Children. This means we are focused on school environments and the cities that you can see on the map here. At Aarhus University, we have been responsible for a task about the development of protocols for harmonized approaches and high data quality. Next slide, please. These harmonized approaches were developed for the two major measurement campaigns in the project. The Tier 0 campaign included 3 cities marked in red here, or 3 countries to be more correct, and it ran for a whole school year. It mainly used low-cost sensors to measure temperature, humidity, particles, CO2, and volatile organic compounds. It also included additional microbiological determinations. In Tier 1, we expanded the measurement campaign to 7 cities, but limited the time span. So we measured in 2 seasons, and we included both on-site measurements and sample collections that we then took back to the laboratory and analyzed there. And here we analyzed the full set of physical, chemical, and microbiological parameters. Thank you. However, if we use different methods, we get different results. This is certainly true for coffee, where I found this description here, that besides the coffee-water ratio, the extraction time and grind size can contribute to the perception of strength. And the same coffee made using an espresso machine, a cafetiere, or a drip filter machine can produce different flavors and different caffeine content for various reasons. So what is true for the coffee here is certainly also true for the different factors that can influence the results of our measurement campaign. We have a variety of locations across Europe, and this also includes differences in infrastructure. For example, the ventilation in a school or just the availability of power outlets. We cover different seasons and include continuous measurements for a whole year. So these measurements should, of course, be comparable over time as well. We use different instruments, both on site and in the lab. And of course, there's the human factor. We are no robots, and we might have different ways of sample handling, collection, and analysis. So there is a clear need for standardization to ensure comparability. Next slide, please. So for this reason, we have developed a set of 16 standard operating procedures in the project that Paula will present in detail in a minute. They cover on-site and online measurements, different sampling techniques, laboratory procedures, including the analytical methods, quality assurance and quality control, and we've also included a comprehensive SOP on integrated exposure assessment. These SOPs were then used in the measurement campaigns, tested and validated, and revised where necessary. So Louise will present this part of the work. Next slide, please. Thank you. As a related but complementary activity of our task, we developed a walkthrough survey together with the whole consortium for a standardized characterization of classrooms, schools, and their surroundings. It is obvious that schools can differ in many ways. I've just put two pictures here of very different architecture of schools. The upper picture actually shows my own school, which was built in 1530 in Germany. And the lower picture shows the school in Copenhagen, which was built in 2021. For the walkthrough survey, we developed a lot of questions in an interdisciplinary approach, where we followed these criteria. The questions had to be relevant for the integrated exposure assessment, relevant for all cities and countries, and they should be unambiguous with regard to their phrasing. So this resulted in a pool of 200 questions, which we then divided into a set of mandatory questions, 86 of them, and optional questions.